All right. So what we're going to do here is we're going to talk about how we're going to take our genes and we're going to make them into who we are. So it's genotype to phenotype. In this short little one, I'm going to talk about genes to message, but we're really talking about getting what's in your DNA to be expressed as what you see as your body. And what you see as your body is your phenotype, and that's made up of your genotype and the environment and how the environment has influenced how those genes are expressed. So by the end of these short videos that I'm going to show you as far as how we're going to take our genes and turn them into active proteins, you should be able to list the differences between DNA, genes, RNA, and protein. You should be able to describe the differences between genetics, epigenetics, and really what we're talking about again is genetics is just what's in your DNA. Epigenetics is how that DNA has been shaped by your environment. You should be able to explain the components of your phenotype, differentiate between intronic and exonic DNA. You should be able to define a single nucleotide polymorphism, so a small mutation, and how that could change your physical function. You should be able to describe transcription and state how it's controlled. You should be able to explain what a promoter is and how it's controlled, and how you get basically more expression of a gene. You should be able to list the differences between transcription and translation. These are two very similar processes, but one is taking your DNA and transcribing it into RNA. The other is translating your RNA into protein. You should be able to state how much or how the amount of mRNA can be regulated within a cell. So really, this is a balance between synthesis and degradation. So how are we controlling these things? You should be able to list the steps of translation and tell me how they're regulated. And you should be able to state how the folding of a protein can regulate its function. All right, so the first thing and the easiest thing to do is to talk about this idea of genotype and phenotype. So genotype is what our DNA has. Each cell within our body has exactly the same genes in it, has exactly the same DNA in it. But what we look like is the combination of the genes that are in our cells and the environment under which we challenge ourselves. So this is a classic example of Otto and Ewald. They're two German brothers. They're identical twins, but one of them became a long distance runner. Here, that's Otto, and the other became a strength power lifter. That's Ewald. So even though they're genetically identical, these two individuals are vastly different physically. So this is 18 years old. This is them at 18 years old. If you go on later in their life, they take on even greater differences. So this is the difference between somebody who has got the exact same genes, but one who does strength training and one who does endurance training. So the type of training, the type of nutrients, the type of activities we do, whether we smoke or not, that shapes our genotype and becomes our phenotype. So how do we determine our phenotype? An athlete's phenotype is determined by their genes. Can't get away from this. If you want to be an elite athlete, you need to have picked your parents very well. I did not pick my parents very well. They're great for a lot of things. Athletics is not going to be something that I'm going to have a genetic advantage at. How much of each gene is made into RNA is going to be important. So that means basically our epigenetics. How much of each mRNA is made into protein. So that means how we're shifting mRNA into protein. And that's basically what we do with strength training, is we make more protein from the same amount of RNA. When we do endurance training, we actually make more RNA from specific genes. How active the protein is, that is something that we regulate on a minute-to-minute basis. What exercise have we done? What have we had nutritionally? All of those things are going to be really important. And therefore, when we want to understand how to maximize one person's phenotype and their performance, we must understand how to maximize making the right mRNAs, turning those mRNAs into the right proteins, and then activating those proteins so that they can be as highly active as possible.